Our next speaker is Dr. Peter Fedichev, and he received his PhD from the University of Amsterdam. He's a co-founder of Gero, a physics-enabled biotechnology company creating therapeutics against chronic diseases with a mission to slow down human aging. He's an author of 75 plus published papers in multiple domain areas, including publications in Science and Nature Communications.
Thank you very much. It's a pleasure to talk after Steve about also mechanisms that would be controlling average and maximum lifespan, which are different, not only across different species, but also in humans. So Steve has just shown you this wonderful chart suggesting many animals, suggesting that there is at least quantitative difference between different species in terms of the rate of aging. What I would suggest is that aside from that, there is also qualitative difference. I mean, species age in a very different way. So on the left of my chart, I have humans, and just for fun, I didn't do log transform. So this is mortality in humans. It's ridiculous. There is no other known species with such a range of change of mortality. This is this famous Gompertz law, doubling of chances to die every 8 years. And then on the other hand, we have these guys like naked mole rats, which are not immortal, of course, but at least they do not show any acceleration of mortality for as much as you can measure, whatever it means. Either they are aging very slow, or I would like to age as slow as they do, okay? Whether they are negligibly senescent or not, I don't care in a way. So when you see these exponents, and I'm coming from physical sciences, our original idea was that, okay, there are many examples in engineering and in nature where there are systems that are built of the same hardware which could behave in a very different way. You can have a nuclear reactor and then it can suddenly explode because you turn the knob a little bit in the wrong way. So the same stuff, a little bit different regulation, transforms the system from peaceful nuclear reactor of enormous power to a megaton bomb, if you like or don't like. So we had an idea that if we could get enough data from animals and humans, and if we could use modern machine learning tools in order to embed them, how they call it now, or transform them into some kind of trajectories, we could differentiate that maybe some animals, if we could have sufficiently many points of the same animals across the trajectory, that maybe some animals, and we were thinking maybe humans, are of this kind of exponentially unstable guys, exploding. That's called aging. And maybe there are some other guys who are not exploding. And basically the idea was to get enough data. So we started getting data from animals, from mice, mouse phenome database, from our academic friends, from everyone who had these longitudinal trajectories. We got to UK Biobank. We started publishing papers. We got collaboration with Pfizer this year, and they gave us 10 million electronic medical records, and now another pharma company gave us 100 million electronic medical records. And of course, these are not DNA methylations. Of course, I would like to have, I don't know, 500,000 measurements in each of my cells 100 times a second. I mean, that would be a wonderful data set, of course. But unfortunately, nothing like this happens longitudinally. And electronic medical records are the best longitudinal data set in humans. Not the best in terms of, biological value, but it's legally accurate, okay? Because people are paying for those services. So by looking at the data, we observed a bunch of interesting facts, published a bunch of papers to get your attention. Now I got your attention, so I will tell you actually what does it mean, as negative as it sounds. So what do? We use machine learning, which means that we use networks in order to take a health state of an animal today, convert it into some kind of very compact representation, just cast it into a few numbers, and then use those few numbers to predict the health state of an animal or of a human in a few months, years ahead. So interestingly, that when you apply such machinery to mice, it turns out that it is sufficient to get just one number. So the network should know just one number about this animal in order to predict the future state. And that number on the middle chart is going up exponentially as these animals are aging, right? So, I mean, this is an unsupervised model. We didn't put any law there, not linear law, whatever. I mean, we just looked at the data, and the data told us that mice are aging exponentially. So you see there is something that is measured by that number, which is apparently the total amount of damage accumulated, that is going up exponentially with age, and the doubling rate is exactly the mortality rate doubling time, which means that you can measure something in an animal dynamically and see things that are changing at the same rate as the mortality changes, okay? Which means that most probably there is a direct relation between the two. And also I like these red dots. So those red dots are the animals that are scheduled to be killed in the lab because they are too frail okay, by ethical considerations. Which means that in the lab, at least, mice are dying from excess biological age. You can see they reach a certain biological age level and then they're killed. Okay, that's the dominant cause of death for mice in the lab. So these markers that you are getting just from machine learning in an unsupervised way correlate to all hallmarks of aging, even with those that are not in the training set. So for example, on the left, I have the total number of senescent cells in the mice of different age, and then I have the correlation of the same number of senescent cells measured with luciferase, say, as a function of the biological age inferred by the model that was using just the blood cell counts. And we can see that these things are correlated to the biological age better than the chronological age. So actually, these models are smart enough to produce the biological age that is better than the chronological age. So okay, this exponent means that these mice are unstable, which means that we know how mice age. And instability looks scary, but in fact, it has an interesting practical implication. So if you are unstable, it means that you don't know what is your equilibrium state, which means that if I apply an intervention that either reduces the biological age or increases the biological age, the treated group should always be below or above the control. No equilibrium means that I can rejuvenate somebody. So if you're a mouse, I can rejuvenate you. So these are typical experiments that we do with our mice. So you have two groups, then you have a shock treatment, just very large dose of something that extends lifespan in these animals. And you can see that after the treatment, the treated group, this bluish color, is always below the control, which means that, once again, there is a very direct relation between stability and rejuvenation. If you are unstable, it's very easy to rejuvenate. And if you are stable, it won't work. So, okay. You have these networks, you have all this software, and you throw this software at humans. And as I said, there may be, and I will try to make the case for, that there is a quantitative difference between mice and humans in a very important way. So if you make out of what you have a biomarker of aging in humans and plot it as a function of age, I mean, first of all, you would see growth, right? I mean, for the first 20 years, your biomarker would change, but that's definitely not aging. Just for those who care, there is a wonderful book and Science and Nature paper by Jeffrey West from Santa Fe. So for the left part of this curve, for the growth, there is an analytical equation. So this part is totally understood by the theory. And we just checked that with blood samples, you can actually reproduce Jeffrey's result. But then, let's say after year 40, you have deviation from the steady state, and those deviations are not exponential. Of course, you can fit exponent there, but the exponent would have 10 times less 10 times more doubling time. So these changes are sub-exponential, which means that you cannot measure anything in humans that is going up exponentially with age longitudinally in an individual human. You have a paradox, essentially. You have exponentially increasing chances to die, and there is no factor that is going up exponentially at the relevant rate. Which means that we are now in a very bad shape because we are acting against molecular factors pharmacologically, and we don't know which molecular factor is responsible for this functional shape. So we started looking at other things. So just we've heard complaints that if you order biological test results today, okay, if I do it today and send it to the same company, I will get most probably the same result. But if I do it 1 month from now and I send it to the same company, I won't get the same result. So what happens is that biological age, like on this plot, is not going up like, in these nice steady curves, like cross-sectional data may suggest you. It's going up on average, but it fluctuates. And the fluctuations are actually higher than the change in the mean with the function of age. So if you look at these fluctuations, you start observing that these fluctuations, if I measure biological age today, the biological age right now, they're correlated. So the longer is the time difference between the measurements, the less is the correlation. So the biological age forgets about, I mean, that's what we all know. If I have an infection, if I have a bad day, in a few days, in a few weeks, I forget about that. My organism recovers. So humans are stable. The biological age decorrelates. And if I measure the rate of the decorrelation of the biological age, the rate, how quickly the body forgets about interventions, you can see that the rate is going down linearly with age. The rate is going down means that the time is going up, which means that the older you are, the longer it takes for you to get back to the norm. And this is an interesting biomarker of aging. You may ask me, okay, I mean, why autocorrelation functional biological age? What the hell is that? But this is what is happening with you all the time. Your biological age is fluctuating, and you can actually study the properties of these fluctuations. If you don't trust me, you can measure the autocorrelation time of your biological age in a hard way. You end up in a hospital for whatever reason, and then you have inverse hospital stay. And okay, in rich countries, people don't stay in hospitals for fun. It's expensive, which means that they're in a bad shape there. So if you look as a function of age, the inverse hospital time, it totally repeats that curve, which means that the autocorrelation time of the biological age is your recovery time, right? So you can really dynamically, from a healthy animal, measure a quantity which would tell you how long would that animal survive in the hospital. So, okay, one thing, we have fluctuations. We don't have growth, we have fluctuations. We don't have exponents. Okay, why do we have the Gompertz law then? So I think it's very easy to understand. If you have longer recovery time, it means that if you are kicked out of the equilibrium, if you have longer recovery time, you would spend more time out of the equilibrium, which means that the range of your fluctuations would go up, right? So we measured one of the variants of the biological ages, different biological ages, pheno age, our biological age, physical activity from wearable devices, something else, I don't remember. So you can see the inverse variance of all biological ages hits zero at about 120 years old. 120, 150, you cannot tell. Exactly at the same point where the recovery rate is hitting zero, which means that this is the same process. So what happens as we age is this. You have any parameter you like, biological age, C-reactive protein, whatever you like. And this is this horizontal axis. And this parameter is fluctuating because life is hitting you all the time, bad sleep, bad president, whatever, bad boss. The things happen, right? So as things happen, your biological age responds to that. So don't complain when you send the samples to companies and get different results, because some days are good and some days are not. The problem is that as we age, our regulatory system is changing, and I will tell you why and how, in such a way that the regulatory potential becomes shallower, which means that it takes longer to respond. The range of the fluctuations is increasing, and on a specific bad day, you lose the stability and start disintegrating. You can be aged and not frail, you can be aged and frail. So aging is the change of that shape, and frailty is the phenotype that you start developing once the stability is lost. So surprise, we were thinking that we will get lots of data from humans, from animals. We'll find that humans and mice are exponentially unstable, naked mole rats are stable, and it turned out that Charles Darwin is working in mysterious ways, gentlemen, ladies and gentlemen. Humans are actually born as naked mole rats. We are stable. Over time, our stability properties are changing in such a way that late in life we lose stability. Late in life, humans become mice. And that's why most of the drugs that we're testing in mice would work in late life humans. So I would say that the deviations of the organism state that are captured, okay, I'll comment about that. So you would tell me, okay, this is a nice theory that probably explains everything, describes everything, but explains nothing. Interesting theory, but what would we do about molecular factors? Do we have a chance to control this? And now I'm going back to methylation, because we've seen methylation data sets, more than 5 years. But we didn't publish anything on that because we couldn't make sense out of them. So if you take any methylation data set in humans, you would find, if you cluster them, lots of features, lots of methylation sites, it's incomprehensible. So if you cluster them in such a way that the same sites behave in the same way, you find that there are few clusters. The more data you have, the more clusters you have. But whatever you do, there will be one cluster of features that change their methylation patterns linearly with age. Okay? It's always there. That's why you can do chronological age, you can predict chronological age. And then, the more data you have, the more clusters of features would have hyperbolic dependencies, like clearly non-linear dependencies. And that's why clustering actually differentiates between them. Okay, so what? If you look at those hyperbolic features, those that are visually non-linear, if you measure the inverse variance of those features, the inverse variance of at least one feature, or always of one feature, will hit zero at about 120 years old. So this is our old friend that we have just observed in blood and in physical activity. And later, spoiler alert, I will tell you what is that medically. So okay, so this is just the same biological age as like Grimm age for Steve. So this is a property of of biological clocks that are more or less like Grimm age. But then, what is this linear feature? Okay, so we have Grimm age that is kind of hyperbolic, whatever, looks like it loses stability at about 120 years old. So what is this linear feature? So let's look not only at the mean of the linear feature, but also at the variance of the linear feature. Both of them are exactly linear. And for those who is mathematically sophisticated, I mean, this is not really sophisticated, this is a signature of Poissonian noise. This is the worst thing that could happen. I mean, this is a compound effect of infinitely many uncorrelated events. That's why you have exactly linear mean, right? I mean, they are uncorrelated. It's damage. I mean, that's called damage. These are things that happen in you in uncorrelated way, separately, each of them with low probability each. But since there are so many things that can go broken in me over time, the total amount of damage is going up. So the linear age in humans has very simple physical, biological, medical, whatever meaning. It's just total number of bits of damage accumulated in you over time. This is your biological clock. So now, think about that. So you have many, many systems. So every system here is like this square with 2 dots. And all the systems are born in a more or less healthy state, in this white state. And I just arrange them by the ease, how easy it is to break them. Okay? There will be some soft features that are easy to break. Okay? You can get metabolic disease once alive, right? I mean, this is a soft feature. And then you have some kind of tonic features like your mutations and everything else that are not allowed to change, right? I mean, they have low probability of changing. The total number of those features is so large that over time they break and the total number is going up linearly, and together each of them is nothing. But together they produce stress on soft features. And they start destroying, they linearly stressing all regulatory interactions for the linear features. They destroy regulatory potentials in a linear way. And so that's why the averages are following the damage accumulation. The recovery rate is following the damage accumulation. But what's most important is that the height of the barrier that is protecting us from disintegration is also going down linearly with age, and this is sufficient to get exponential acceleration of mortality. So all hyperbolic features that you see in your signals are adaptive features. All linear features that you see in your signals are the driving features. The catch is, however, is that the adaptive features are reversible. They are stress responses. They are built evolutionarily to activate and deactivate. And the linear features are Poissonian irreversible. Bad. So targeting so there was a question on this forum, like targeting of those adaptive features may be wrong. Yes, it's wrong if you are stable because they are protecting you. But once you lose stability, forget about protection. You just need to stop them. So that's why most of the interventions against hallmarks of aging actually work. So what we do is that we're taking now these millions of electronic medical records. We encode them into these functional units. So we make AI actually separate all the health, classify in a bunch of states of functional units for which we don't know the meaning yet. And then, since even if you have, 400 diseases in electronic medical data, most of the biological states you do not see. So to represent what we don't see, we introduce one more variable, which is this biological age. So interestingly, the biological age is the total amount of damage in you. So you mostly don't see it in the electronic medical records. Yes, I'm quitting. And then we use those variables to predict the future state, right? We run it on very large data sets. We extract biological age. We extract the probabilities of failures in these functional units. We find one functional unit that has exponential acceleration of chances to fail. This is metabolic health. Who would get surprised at this point? But what's most important is that now we have these functional units and aging nicely separated within the system, and we can use all those phenotypes in order to produce whatever, genome-wide association studies, proteome association studies, whatever, to find targets that separately affect each of the phenotypes. So here I produce GWASs for all our phenotypes from the model, and on the bottom you have GWAS for parental longevity. This is longevity GWAS, and you can see that most of the signals that we see in parental longevity GWAS are actually the signals from diseases. So nicely, this kind of physical intuition actually allows you to separate aging from diseases. So that's why we're making a bunch of predictions. We believe that hallmarks of aging are not relevant in humans. We believe that you can rejuvenate mice. It's hard to rejuvenate a human. We believe that there will be other drugs that affect maximum human lifespan, as Steve is also suggesting, totally other drugs that are affecting maximum human lifespan. That needs to be worked out, and that's what we are working on. Most of the industry are working on these late life phenotypes, hallmarks of aging, and everything else that will help people late in life. And in order to dramatically extend human lifespan, we have to do something else. We do this research ourselves and in collaboration with our academic partners, with Brian Kennedy, Andrei Gudkov, and others. We work with pharma. We regress out aging for them to make sure that they are not developing drugs against aging, right? We help them to develop drugs against not aging diseases. And for fun, we also deploy, you don't need to pay $500 to us. We're also deploying free for all, no user value application that reports biological age estimate for iPhone users. And we're using it in order to observe biological age fluctuations in large cohorts longitudinally, just to learn more about biological age and progress in the science. Thank you very much.
Excellent, Peter, excellent talk. I really, really enjoyed always speaking to you. I was just wondering, I'm trying to wrap my head around this very interesting, complex angle and maybe summarize you and the previous talk, which is, if we think about the methylation clock, then this is a lot like scratches on the windshield. So the scratches themselves do not mean anything, but it's rather the accumulation rate that means something, right? And the older we get, the weaker our systems get that prevent these scratches from happening and that sort of improve them, right? So what you see in your fluctuations is, in any metric that you measure, is that the homeostatic systems, basically, they don't work as well anymore, right? For whatever reason this is.
Yes.
And now if you plot it against the maximum lifespan of any species, well, of course, for an animal to make it that far, these systems need to be more robust.
Yes.
So the more robust they are, the longer the animal can live.
Yes.
So I'm wondering from this angle, maybe the best biomarker for aging isn't a specific metric at all, but it's more of a perturbation, right? So you give a person something hormetic that causes them to,
Yes.
and then you measure how well
Yes,
do they
exactly. That's
contribute.
what I think we try to demonstrate. If you can measure resilience, or your inverse hospital stay is a wonderful biomarker, but this is also somewhat inhuman.
Yes.
So that's why we suggested an alternative measure. So by observing the properties of fluctuations, you can actually infer the resilience, and you can estimate the robustness of these systems. If you let me maybe to relate to the previous talk and regarding your comment, I find it very interesting. Steve reported that this development time correlates with longevity and maximum lifespan through metrics. I always like to think about it. So when you grow, you always have this investment decision. You should invest either in repair or in growth. If you want to grow quickly, you underinvest in repair and you are a mouse. So if you care to invest in repair, you become human and make it more red. So interestingly that this resilience is actually measures your investment in recovery. So I think that in some future, when we will have enough data on all animals, longitudinal data, methylation, genetics on different species, we will see lots of these correlations.